are you doing? Welcome. Welcome to the, let's call it wrap-up lecture. We will talk about transmembrane prediction and mostly about disorder. To repeat, you can predict secondary structure by putting in a consecutive window of, in this particular case, 13 residues that are coded such that each blob here is standing for 21 units, 20 amino acids, and 1 spacer. You predict secondary structure in 3 states, HEL. You could also, okay, let's get into that one. Anybody of you familiar with SVMs? Was that a yes or was that just, nobody's familiar with SVMs? So SVMs are in principle like neural networks. They project the input into a higher dimensional space, and then they try to separate the points in the higher dimensional space. Now, the major difference, there's one major difference, and there are 2 implications of that. So, trying to get the chalk. So, say these are points, and you want to classify the yellow and the red. So in a high dimensional space, I'm going to draw a line, but in principle this is a hyperplane. In a higher dimensional space, the neural network finds a line such as this one. Sometimes it makes mistakes, so the line would go like this. And there are many other lines that are compatible with the complete separation of the problem. Now, the point is that the neural network actually considers all the points. And there's no weighing, so they have equal importance, all the points. While the SVM zooms into points that have different labels, red and yellow, and are close. And then essentially it argues, well, these points here, they are trivial. So let's just throw them away. Maybe this is trivial. Maybe these 2, I don't know. And let's do the separation only on whatever is closest. And there are 2 implications for that. One, you can get away with much less data. Because typically, in fact, the vast majority of points sit somewhere else, somewhere here. It's relatively easy to classify. In the case of secondary structure prediction, we see this because relatively simple devices, relatively simple statistical devices can already separate at a 55% accuracy. So the neural network adds something like 63 without taking evolutionary information. But there's some part of the problem already solved. And these points essentially are thrown away by the SVM, and therefore, at the end of the day, it uses fewer points. Is anybody in the room here able to see what the problem of that could be? So again, it zooms into finding whatever points are labeled differently, yellow and red, and are close to each other. Can anybody suspect what the problem of that could be? So the great aspect is, essentially, you need fewer data points. Yeah?
You don't get the whole picture. And
that is true.
Those points are just miscategorized data.
That's the major point. So the first point you made is true. And that could be a problem, but usually you don't really care because you believe that these points are classified anyway well. It could not completely give you the right distribution. It could go wrong. That's true. But the second point is much more important, which is, since it zooms into the ones that are most similar to each other and differently labeled, it somehow is like an error searching device. If you made a mistake in the experimental labeling, then these are the points that you're going to find, right? So if that should have been a yellow point in that one too, then the device has an intrinsic problem. It tries to separate out 2 points that in fact are not separatable simply because they stem from an error. And these are most likely exactly where the things are very close. So somehow it's a device that zooms into mistaken or incorrect data, noise in the data and the labeling. And therefore it's very good if and only if you have really accurate data. I told you, here's the other point I wanted to make on this slide. Now, the neural network, I described the way it works. you can have an arbitrary number of output units. In the SVM, you can actually not. It's a separation of two classes. It's essentially a binary problem. In this particular case, yellow and red. How could you transfer this problem of predicting second-order structure in 3 states onto a binary problem?
You can do 2 times binary.
Exactly. You could have one SVM that says helix against the universe and beta strand against the universe, or possibly a third one. So there are different ways in which you can play this. And then you have to find some mechanism by which you decide, okay, it's predicted to be helix and strand, what are you going to say now? You could argue that you take whatever is the highest, or there are different solutions for that. Okay, but it's slightly less trivial than the neural network in that sense. I told you that with this device we do much better than the first and second generation, so generations that used only one amino acid, that used only segments statistically. The machine learning, bless you, improves because it can specifically train better on beta strands. It can specifically be designed such that short segments are no longer a problem, and we can put the evolutionary information in, and then it really shines. We talked briefly about transmembrane helix prediction, and I'm going to try again with a movie. It's one of those things. It's the show effect. I made it work on 3 projectors with 2 different machines. Oh, it's going. Okay. So then all we need is the sound.
The cell membrane controls.
Okay. Thank you for spotting that. Now I didn't get the sound right.
The cell membrane controls traffic into and out of the cell surroundings. Like all biological membranes, the cell membrane exhibits selective permeability. That is, it allows some substances to cross it more easily than others. This ability of the cell to discriminate in its chemical exchanges with its environment is fundamental to life, and it is the cell membrane that makes this selectivity possible. In this video, you will learn about the structure of membranes, how the outermost membrane of the eukaryotic cell, the cell membrane or plasma membrane, controls the passage of substances. However, the same general principles of membrane traffic also apply to the many varieties of internal membranes that partition the eukaryotic cell. Membranes are found around the entire cell: the mitochondria, lysosome, nucleus, and many other organelles. Membranes serve to separate the contents of a cell or organelle from its surroundings. Here you can see a lysosome, an enzyme-containing organelle, as it is digesting an old, worn-out organelle. The membrane surrounding the digested enzyme is necessary as a barrier between the digested enzyme and the rest of the cell. The cell membrane is made up of 4 main components: carbohydrates, cholesterol, phospholipids, and proteins. Lipids form a barrier that separates the inside of the cell from the outside of the cell. The phospholipids are arranged in a double layer called a lipid bilayer. A pure phospholipid bilayer would allow a very limited number of substances to pass through. Water can be fused across because it is so small, but this is a slow process. Each phospholipid is made up of a phosphate group and 2 fatty acid tails. The hydrocarbon tails are non-polar, unable to bond with water, and are hydrophobic. Hydro, water, phobic, fear. The polar phospholipid head has an affinity for water, and thus is hydrophilic. When phospholipids are added to water, they form assemblies to shield their hydrophobic portions from the water. See here the formation of a lipid monolayer.
Okay, and again, the way you transport material through the cell wall, the point was here water can diffuse through. but active transport of ions and all other material in and out is regulated by proteins that sit in the cell wall and let specific ions, for instance, through. And these, in some sense, are the gatekeepers. That's why most drugs act on gatekeepers. That's the message of this slide. About 57% of all the drug targets are membrane proteins. Transmembrane helical prediction, since it is hydrophobic, inside you look at the hydrophobicity scale, and one way to predict transmembrane helices is you draw a hydrophobicity scale. Higher values here mean higher hydrophobicity, more likely therefore to be in the membrane. And then you somehow look at a threshold, a cutoff in this, and say everything above that threshold is a membrane helix. Membrane helices have to have a certain length in order to get through the membrane. That typically is in the ballpark of 20 residues, because membranes, so unlike in this drawing here, membranes have a fairly constant thickness. It's about 3 nanometer in the ballpark, 2.5 to 3.5 nanometers. And that's around the entire cell, the same essentially. So my sausage drawing here is not quite right. And that implies when you want to go orthogonal through the membrane, there's a certain length you need in order to cross it. And that is in the ballpark of 20 to 25 residues. So the hydrophobicity should be high for this entire region that goes through it. Now, how can I possibly distinguish between the outside and the inside? So whether the tail of the membrane protein starts here, or like in this case here, starts here. I want to know, because for the type of protein that I have, this is a difference. So sometimes the number of membrane helices and the orientation, so where it starts, is exactly what sort of describes the function of the thing. And there's a rule that when you look at cytosolic, and the inside, you have more positive charges here in the inside than on the outside. So what I'm showing here is the red versus the blue. And you see the red is more positively charged than the blue. And positively charged here simply means you count up the residues with positive charges. So now if you want to predict a membrane protein, one way in which you can do that is you simply predict the membrane helices through the hydrophobicity, and then you simply count how many positive charges you have here, how many positive charges you have here. Inside you have more, so whatever is more gives you the direction. Again, for the membrane prediction, you can also use the evolutionary information, put this into a neural network just the same way as before, and the difference being that we now have 2 output units. In this particular case, remember that I had a second level network. So the first level network used sequences input and predicted secondary structure. The second level network used predicted secondary structure and then predicted secondary structure again to see that LHL doesn't exist. So helix has a certain length. And for the membrane residues, this is even more important. The problem for this particular case here is that so the first level is it gets only the half length of a typical membrane helix. The second layer gets twice the length. So somehow this is a clear overshot in this particular case. But first hack was to just hack them apart. And the second hack was a slightly different idea, and I want to just illustrate that. The idea is, so the 2 units transmembrane knot, along here runs the sequence, and essentially they are one minus of the other one. So this is the unit for transmembrane, this is the unit for not transmembrane. You see when this one not transmembrane is up, the one for transmembrane is down, and vice versa. So now you can perceive this top graph here as an energy landscape, or as some function, and you want to optimize the path through that function given certain constraints. Constraints are, for instance, that transmembrane helices are between something like 20 to 25 residues long. So given that constraint, find me the optimal path through that landscape. And if you begin, so the first step is you would simply label the average prediction for every single possible transmembrane helix. Okay? This is the pool up here. Now, which one would you choose? If you believed that the protein, if you want to predict the first transmembrane helix, which one of this pool would you choose here? So in the first assumption, now you have the pool of possible transmembrane helices, and you see they overlap. So again, here runs the sequence, and this is really meant to be the position. So these three here, for instance, overlap. Inside the box here, you see the average propensity for being in a transmembrane helix. Now, say I believe, I want to bet that my protein has one membrane helix. Which one would you take? Of all of these up here. It's not trivial? Is it too trivial or is it not trivial? Rephrase. If you want to make a bet, you have numbers up here. What's your best bet? Yeah?
Best number?
And that is too trivial, right? So nobody says it because it's too trivial, is that right? I assume the answer is thank you that you dared saying it. In this particular case, the trivial answer is really right. So now, the second highest, that would actually be this one here. If you now believed it has two, the second highest in my construction is this one. That cannot be, because they overlap. Okay, that is already taken. So if you really believe it has two, then your best guess is to take the second best that is far away from here. So that would be that. And you keep going. You keep going until the signet disappears. So adding another transmembrane helix gives you less of a signal. So in the energy landscape, you reduce the overall score, right? So you fall, for instance, below 0.5. And which means that not transmembrane helixes, the probability is higher there. Now that means that for the assumption that you have three membrane helices, you simply found the best path through that without explicitly saying membrane helices have to always be 20 residues long. You just find the optimal path that is locally optimal for that length. So some of these helices that you construct this way, so again, you have to have two constraints. But they can be fairly wide. So you could say sample anything between 15 and 30. Or some number, right? You can make that up. And you see that you get immediately a relatively optimized prediction, and you get something on top, and it's totally trivial. Something on top of what you did before. All you needed are two constraints. So you could not do that if helices are between 1 and 100 or something like that. You need some additional constraint to go through that landscape. And then you do as before, you predict the topology, and when you do this thing, then this, in this particular case, is the prediction that you get out, so very accurate. Another method in machine learning that is very well to handle a problem like this one here is a transmembrane, is a HMM hidden Markov model. The point of the hidden Markov model is you essentially build a probabilistic model that considers different states. So you begin in some globular state, you're next to a loop that is next to the transmembrane helix. Those are the helix regions. You can model the end of the helix, where the helix actually sees water, the cap here on both sides. You can model the helix core, where it just sees lipids. And then you go into the inside, and there's a variety of states. Model here means for every transition, the hidden Markov model, just like the ones for the sequence alignments, you set a certain probability. Initially, how do you choose these probabilities? If you want to build a model like that?
Take it from your database.
So essentially, you look up in the label that you see in the database, what is the background probability? Let's call it prior, let's call it simplest probability, let's call it overall number from the database. It's all the same idea, right? You look it up from the database. And you look it up according to these states that you create here. So where a state could be inside helix core, or a state could be the end of a helix. And now once you have these, then you can begin to really train particular transmembrane observations that you have. So labels, experimental labels. So the first one, the priors, is just the entire database. And on the training level now, you run all the transmembrane proteins for which you have the detailed levels and optimize these parameters, such that the model fits, and then comes a new sequence, and you simply go through the path of highest probability. And in this sense, the hidden Markov model really is very, very well suited to the problem. When you predict transmembrane helices with the variety of different methods shown here, then so these are very different methods. One of those I showed you is this one here. TMHMM is another one I showed you. That is the green one here, the hidden Markov model. The score is the percentage of proteins for which the helices are correctly predicted. There are some differences, and there are some similarities, and there are some best methods. Now, if I ran this for the entire protein, for the entire proteome, I can now use this simple tool to ask how many proteins in human are membrane. And the answer is roughly 25 here. So this is human here, the third one, around about 20 to 25%. So now getting back to the reason, the drug targets. So membrane proteins are very important because we need them for drug development. And they're important because they make up about a fifth or fourth of our cell. And structure determination for those is extremely difficult. That's why we need very good prediction methods for that. I talked about helical membrane proteins. So proteins that go through the membrane with a helix. Here's another type, and Henry Bigelow, the slides that I show come from Henry Bigelow in the work. Those are so-called porins. So in these porins, you go through the membrane, so the lipid bilayer is shown here by these dashed lines. This is the outside here, up here. And you essentially build up sheets. So this is one strand, another strand, another strand, another, so they form sheets at an angle of 45 degrees. And this way, they really, seen from the top, they make a hole into the membrane. These are, when Henry worked on that, this was a complete set. Sorry, the complete set of all beta membrane proteins that existed. So then there were 12. Now again, you can build a hidden Markov model where you model different states. So the strand goes up, the strand goes down, there's something inside, there's something outside. And this, in fact, is the resulting model that appears to fit the data. The problem is when you have only 12 experimental data points, it's very difficult to see whether you're right or not. Another feature of 1D structure is solvent accessibility. The idea is and I'm a little bit, so this is my residues A, B, C, D, E, F, G. So the protein is somewhere assumed to be outside. There's water above. And so one way to compile solvent accessibility once you have a 3D structure is you just roll a water ball over the protein and see what angstrom, square angstrom, is accessible to water. Okay? And the solvent accessibility of residue B is just defined by the amount of water ball that you can roll around it. You cannot roll a water ball here because it doesn't fit. You cannot roll a water ball here because there's another residue sitting. And this way, essentially, you can classify residues according to how much exposed to water they are. You can scale this in square angstrom. You can use that in a percentage. So there's a maximal surface that every residue can access. And you can talk about the percentage of maximally exposed. When you do that, whether a residue is 100% or 80% exposed, it's almost the same, they're exposed. If it is 5 or 0, that's a big difference. So in terms of number, one of the problems here is the numbers 100 to 80 is 20 percentage points difference, while 0 to 5 is 5 percentage points difference, but the one 0 to 5 matters much more. And that essentially is because 100 or 80, they are essentially totally exposed. 0 to 5. 0 means there's absolutely no way water ever gets in there. With 5, sometimes there may be a position where water actually intrudes. And that is the difference. Now, if you want to map this problem into a machine learning device, so now you want to take a sequence, again, just like we did before, and you want to predict solvent accessibility. How do you code for solvent accessibility? What could you use? There are many ways. Just throw some, give me some ideas or suggestions. It's really easy. You want to predict solvent accessibility. So what is it that you're going to measure? Or what could you predict? What could you measure? Nobody? Nothing? You could measure the square angstrom number, right? The square angstrom number has a problem. You could measure the percentage. The percentage is a way of doing it, but it has a problem. That is the problem that I show you there. 100 to 80 is less important to separate than 5 and 0. So if you translate the percentage into a raw number will not reflect that. Another way in which you could do it is you could introduce states. You could say, okay, what this implies somehow, there is an uneven distribution about importance of the scale between 0 and 100. So maybe there is a point where I can say it's exposed and buried. So then I have two states, ideal for an SVM. But there's another way in which we can do it. We can simply come up with a function that somehow approximates what we perceive as important. And here is one idea, one solution. You use a square root. In the square root function, you take the percentage solvent accessibility and you project it by computing the square root onto 10 different states. And then you essentially predict these 10 different states. Historically, it has been done by just looking at hydrophobicity. Hydrophobicity is the degree to which you want to be inside of a protein. So turn that around and you can predict solvent accessibility with that. Or in this particular case, again, you can use a neural network that uses, again, evolutionary information, and in this particular case is 10 output states, where the coding of the output state here, this is solvent accessibility percentage larger than 81, 64, and so forth, so this is the square number. That means, again, that for the difference between 0 and 1, you have as equal number of units as between the difference between 81 and 64. And that is exactly the idea. You could try to change the way you measure solvent accessibility. I asked you how you could do that. Those are different numbers. And in fact, over the time, people tried them all. Here's another idea. The other idea is you simply use the way in which you define solvent accessibility that is most easy to predict. And again, in the application of machine learning, that idea is sometimes relevant. You need something else here. Easy to predict, you have to always remember what is random. You have to be careful that you're not, for instance, if I look at a 3-state problem, H, E, L, and I look at a 2-state problem, H, versus the rest of the universe, then one has a much higher random probability. One is 50-50 essentially, and one is 1 in 3, so 33. And you have to account for that. When you talk about predictability, you have to always somehow consider random. And typically that's not easy. It's very easy to say, it's a very easy directive, but typically a lot of work goes into this. And again, I'm just putting up something here that has been used for solvent accessibility, but it's a general idea of doing machine learning for any problem that you encounter. Okay, let's go into something IDP. So we have IDP projects available.
About what you said. So you just said, if I'm correct, you just said like whether we can predict a structure from a sequence of a protein depends on the protein. So what are the factors actually?
So I believe I said something that is very confusing. It is true. So can we predict protein structure from sequence? Statement number 1. Yes, it depends on the protein whether we can do that or not. Very short proteins, we can predict. 99% of all the proteins are too long. We cannot predict. For the ones that are too long for molecular dynamic simulations, we can still do comparative modeling. That's only for 50% of the proteins. But for some proteins, we can do that better than others. So it always, in the world of biology, it always depends on the protein. Some are easier than others. But what I just now try to say is that in defining whatever goal of prediction that you have, try to see what is a random prediction in the background. And that does, in fact, depend on the protein, but that's not what I meant. I meant it depends more on the way in which you phrase your task. if you phrase your task as, I have to predict helix versus other, or if you phrase your task, I have to predict helix, strand, other, 3 states versus 2 states, the random probability of these two is very different. So in this idea that I put on the whiteboard, in which you try to define solvent accessibility as a goal such that you can predict it, you have to be always careful. If you have a 2-state prediction, you may argue, well, my prediction accuracy is higher, therefore it's easier to predict it, therefore it's better to predict it. That is always with respect to random. That's all I meant here. And again, so let me repeat that point. We have IDPs available. One person you can contact is the one who's doing the slides. Thanks, Dimitri, and he's working now. But now this project here is about, thanks, Dimitri, intrinsically disordered proteins. What I have said so far implicitly was a mantra, if you want, that always said structure determines function. Sequence determines structure. You take the sequence, you throw it into the solvent, it adopts the same 3-dimensional structure, and that is relevant because the detail, the intricate details of the structure are relevant for the function. That's a very, very simple mantra, and this mantra falls into kinds of shapes in which you essentially see shape codes for function. Shape is relevant. You violate shape, you often get diseases. Now we get into a new world in some sense, or not. That is a little bit of a debate, but the world is such, and maybe this is not the ideal way of looking at it. So in the regular world, you have trypsin, you have an inhibitor, the trypsin fits to the inhibitor, it's the key and the keyhole, things fit together. So you have to maintain shape to maintain binding. But maintain shape, part of maintenance of shape has to do with dynamics. So regions where you can move in order to do a certain swing move in certain binding mode, in fact, they sometimes really have to be very dynamic. But that's also built into the structure. It's part of the structure. And that is also, when you put the sequence into solvent, you get the same 3-dimensional structure. You get also the same dynamics. Again, that's all essential. And you can use that to predict B-values, for instance. Now, B-values are values that measure experimentally the degree to which a particular residue is likely to move. In this figure here, so there are regions that appear to be more flexible and more movable than others. So can we predict this? Some of you may remember Predrag, Rivoyats, and in this particular case here, in this coat protein, the question is, where is this protein movable? Where, if I could predict from sequence where it moves, then maybe I could predict something about function. And here's another example. When we look at the overall distribution of these B-values, it's a sort of normalized B-value, that's a frequency. That means very rigid, and that means very flexible. And then there is some distribution. There are different curves here. What the difference is doesn't really matter. Essentially, they are very, very similar anyway. Now, if you want to predict B-values, the first question that you have is, where do you put the threshold? So, for instance, back to the story that we had with solvent accessibility, actually we could again go for the absolute value. It's not easy. And that was also one of the problems why we cannot do ASA easily. Typically, machine learning, think about an SVM, you need 2 states. Real numbers are not that easy to code. So if you wanted to put states, where would you put the state threshold? Can anybody, I throw you the pointer and you put a state? Yeah? At 0. So when you say at 0, you mean this point here that is slightly right of the peak. Is that what you mean, or is it, or I'm just asking. I'm not implying anything. Don't answer in any way that I don't want to influence you. I just want to understand. So 0, because the gray line here in the background are not easy to see. The peak, I believe, here is at minus, well, roughly -1, right? So given that the peak is at roughly -1, you say 0. Is that right? If so, why?
It's just an idea.
Yes, perfect. It's an idea. So whoever feels this is a good idea, tell me why this is a good idea. And if you don't feel it's a good idea, tell me why not. Let's do it differently. What about putting the peak, the threshold, into the peak? How would you like that as an idea? Yeah?
Another question just for understanding the chart. What does frequency on the y-axis mean?
Number of occurrence. So essentially what we are looking at is the normalized B value for a particular residue. Now I'm asking for a large set of proteins, thousands of proteins. How many residues do I find with a given B value? Okay? And the majority of those are somehow a little bit more rigid, is what it says. But then there's a longer tail on this side. So it's like Tolstoy in some sense. There are different ways in which you can be flexible. There's only one way in which you can be rigid. There are different ways in which you can be flexible somehow. That makes the tail a little bit longer here, right? Most residues, the mass of the distribution sits here. So the mass of the distribution sits on the side of more rigid. But some residues are flexible and some are very flexible. Okay? Now the question again. What about the idea of putting the threshold? I want to predict whether or not a residue is flexible. And I do not believe that I can actually predict this number here. I believe this is too complicated. But I do believe that I could predict a sort of binary value. Flexible, not. That I believe. So I hope that I can distinguish between residues that are somewhere here in the very extreme and somewhere here. I cannot put, let's put this point out here. I cannot put the threshold into -2 and +4 for development because the data is a little bit too small. That would be an extreme way, and it's not even, if you had enough data, that might be the best way of doing it. But we may not have enough data for that. So we can exclude something, but not just take this part and this part. That is not enough. Again, the question is, what do you say if I propose to you to put the peak in the middle in the Gaussian? Yeah? In the distribution, yes?
So what is exactly for B value being 0?
So the B value 0 does not quite really have a meaning. Below 0, we tend to be more rigid than expected, or more rigid than average, or something like that. So lower than 0 tends to be more rigid, higher than 0 tends to be more flexible. So 0 could, in fact, the only proposition from somebody in the room on the table is anyway 0. And 0 is sort of the biological interpretation. So it makes a lot of sense. But my question now is, what do you say if I put it into the peak? And say the peak here is we're at -1. I cannot quite see where it is. Let me just postulate that the peak is at -1. And I say that less than -1 I call rigid, more than -1 I call flexible. Is that a good idea or not? Can you argue? And again, in an exam, in some sense, as long as you give me a clever argument, either of those two, you can answer either way. Just give me an argument that is not nonsense, and that you really believe in, yeah?
If you are trying to compute the flexibility, then it should be more towards the flexible side.
Yes, so if you want to if you care more about flexible, then you want to be more on that side. If you care more about rigid, you want to be more on that side. Back the question. What about this choice of -1? Yeah?
I think it's a bad idea, because if you imagine that the highest frequency was at -2, for example, and we would then choose the threshold at the peak, we would divide it really poorly, because everything to the right of -2 is still pretty rigid, but we would predict it as flexible because we have our threshold at the peak. So we relate our threshold to our research data.
Okay. So let me repeat that point. I smiled at you because I thought you had the solution. But you just got in a slightly different direction. But the point here is, So keep in order to understand this point, you have to keep in the back of your head that I said, so biologically it's larger than 0, smaller than 0. By putting the point into -1, since there is a peak, you somehow violate the biological intuition of the story for many, 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 because between the -1 and the 0, there's a lot of data points. And I would this way make a mistake for many data points, and that is certainly true. That's a very important observation, and that is already a sufficient argument to say for arguing that -1 is not a good solution. Yeah?
What do you mean by biological value is 0?
Yeah, no, I didn't say logical. I said biological.
What do you mean by this?
So that is the people who do these structures. They look at the structure, and then they look at these values, and then they often see that when they look at regions that are more flexible, then they're typically more than 0. And then regions that are less flexible are typically less than 0. It's sort of an intuitive classification that you arrive at when you look at a lot of structures. But again, let's just get away from the biology. And so this argument is right, there's something to it, but let's get away from the biology. I'm dwelling on this point because it's relevant maybe relevant for the future. Yeah?
I mean, just let me understand, we are predicting whether protein is more rigid or more flexible.
We want to predict that. Mm-hmm.
So it doesn't really have something to do with the occurrence, whether there's more, where is the peak? I mean, it doesn't have really much to do with
Let's go a step back. This is not quite true. So I have to say something else. We're not predicting whether the protein is flexible or not. We're predicting whether a residue is flexible or not. And whether a residue is flexible, those ones here are flexible. Those ones here are not. So this value clearly has to do something with it. Okay, so there's something in here where I'm undecided, and that's exactly the problem with choosing the threshold, and that's exactly why I said, well, we could make an extreme cut here, 4 and -2. That makes a lot of sense, but we don't have enough data. We cannot do that, yes?
I'm a little bit afraid to ask this because I came late. Can you maybe wrap up the topic we are talking about, which threshold we are talking about?
So now I want to predict to which extent a particular residue in a structure can move. And what I show you is experimental data for the overall movement probability of residues. Things that move on that side of the distribution, things that don't move on that side of the distribution. And over a bunch of, I don't know how many these were, maybe 1,000, yes, I do know how many there were, 1,000 proteins up here. 1,513 proteins to be more precise. Proteins, this is the distribution you get.
And you want to predict right now how they move?
So essentially I want to predict the flexibility of an individual residue. So this is done once I know the structure. Then I know how flexible they are. But can I do that from sequence without knowing the structure? Because I know that movement is an aspect of function that is important to capture. So can I predict it? And it's again, I call it the 1D feature because it can write it down in a string. As simple as that. My question is, when I do build my machine learning device, where do I put the threshold? And the last question that I have is, if I put my threshold into the peak of the distribution, for the sake of the argument, let that be -1, is that good or bad? Can you argue for that? Yes?
Another idea, if we can only get values between -4 and 6, we could simply choose the middle value. So
if we could only, let me understand again, we take only values between -1 and...
because the left side of the diagram stops at -4.
Yeah.
And the other side at 6. So
Yeah.
just take the mean value of -4 and 6. So that would be 1, I think.
Yeah.
And take this as a threshold.
Uh-huh. Okay. Yeah. This is close to what we actually did. It's not that far away. We argued a little bit differently, but yes, we ended up with a threshold like that. But my question again, so that is where to put the threshold is biology. My question here is, we have a peak of the distribution. Why not put the threshold into the peak of the distribution?
Maybe we divide it by using 50% on the one side and 50% on the other side.
Which is close to the peak. Well, in this one is not symmetric, but I believe in this particular case it's not far away from the peak. But my argument is, why is that a bad idea? Or my question is essentially, why is that a bad idea? First you and then you, just a second.
Maybe because the peak is based on this data set. It doesn't really doesn't represent the whole board.
That's true, so be careful. When you always have to wonder whether your data is representative of the universe. And if that gives you a threshold, let's talk about it. But at the point now, we want to put a peak into assuming that this is representative of the universe. So that's currently not our consideration. Just a sec, there's another one here first.
Yeah, same point.
Forget about it. Assume, for the time being, put this question, sometimes you have to put a bunch of questions for your investigation in the back of you. Bring them up once you have solved certain questions. But that is not relevant for this question here.
So, assuming we want to build a machine learning algorithm from that data,
yeah.
We would then categorize the supervised things for 50% that and 50% that, so we could not make a reasonable
Why not? So yes, we would. Again, you guys have a point here with the distribution thing. I'm a little bit rigid because I don't want to hear something else. I want to talk about something else. You got a point there, and this is in some sense is the same point. So if that distribution were not the truth, if that distribution were totally wrong, then I would have clearly to make a different decision here than if this distribution were representative. In this particular case, this distribution is pretty representative of the universe, I'd argue. At least the universe of known structure. So at least for half of all the proteins, I believe this has an important truth to it. Okay, let me bring out the problem. The problem when you put the peak, when you look at any peak of a distribution and you make a decision between left, not, right, yes. So in this particular case, left rigid, right flexible. Then what happens is, well, there are always experimental mistakes, right? So they do a plus epsilon or a minus epsilon here. Tiny mistake. And in this tiny mistake, if you put your threshold into the peak of the distribution, tiny mistakes in the labeling of the data have the highest influence on your training possible, and a bad influence. Because you, for many points, you pretend, say epsilon is the error that you make. You pretend that whether you're on that side of the epsilon or on that side of the epsilon is actually a real difference that you want to machine learn. But if that is just the mistake of the measurement, you don't want to machine learn that. So in fact, you put yourself, by making the cut into this peak, you put yourself into the position where you're most sensitive to random changes. That's always a bad idea. No matter what you do. Now, in most publications, people do exactly that. Whether it's biology, whatever you do, and that's why I dwell for so long on it. It's a very important lesson. Unfortunately, the lesson does not tell you where to put the threshold down. And again, I showed you a few very extreme ones here. Your colleague said put it here. That was one. We are going to put it somewhere here, and actually we're going to have two different thresholds. So to put it anywhere here in the distribution is complicated because here so you somehow have to choose one side. When you choose one side, the way we argued here, the fall is too extreme. So putting it anywhere here is complicated, while here is a little bit more shallow. So it's more likely to put it right here. And we try to put a point where we have enough data on the right-hand side, and we still don't do a big problem. Is that idea, in principle, clear to you? Okay.
How could you choose the threshold there? Do you know the error? How do you know the error from the peak?
So you do not know the error from the peak. You have to compile the error differently. So the error bars are missing here. There's a lot of information I don't show you. So why do I put it here and not here? So we put two thresholds, and by that you already see that we were not sure. Okay? That's one part of the answer. So there is no clear solution. So why did we put it here and not here? And ultimately the way we arrived at it is we moved from this side to a level where we felt that the data, we lose too much data. And that's essentially where we stopped. And we didn't want to get into the top part of the distribution, so we went away to one standard deviation from the peak on that side, and then we came to some point here, and then we essentially optimized according to have enough data points.
So this is something
very empirical.
From practice.
You always do this in an empirical way. What I'm telling you is never put it into the peak. What I'm not telling you where to put the peak. That's where you have to come up with some rationale. And that is where you also have to ask again, how representative is the data? Maybe that helps me in getting the peak. Or the colleague said here, maybe you want to focus on the flexible residues. So you want to put it more on that or on that side. So depending on what you care for most, that has a lot to do with your background, yes?
What is strict mode and non-strict mode?
So this is essentially we train two different machine learning devices, and we train two different machine learning devices, and we use these words for that. In the strict one, so everything it predicted that machine learning device predicted is really flexible, and the other one it sort of is in between kind of mode. Okay, don't worry about the words, but sometimes the statement here is sometimes. Maybe you don't find one empirical solution. And then you're going to train devices on two. And you hope that you can distinguish whether one or the other is better. And I'm not giving you the story about what was better here and why we chose what we chose in the end. The next slide, let me just summarize them all, that you can predict it and you can learn something about function, and this learning about function, there was a cover of a journal here, the prediction method. You can both find things that are important for function and are conserved, and you can find things that are important for function and not conserved. And we're getting back to disorder, and the question here is how is dynamic related to disorder? And what Predrag shows here is that the B factor is not directly proportional to disorder, but there's some correlation. Now disorder, and I realize now that I never showed you the nature of the beast. Protein disorder means the following thing. Typically, you take a sequence, you throw it into a solvent, you adopt a unique three-dimensional structure. Disordered regions are those that you take this sequence, you throw it into solvent, you do not get a unique three-dimensional structure. We cannot do the following experiment, but if you imagined on this disordered region, you now could shine light at different time points, then at different time points you would see different things, different structures. While a regular protein structure, you shoot light at two different time points, X-ray beams, you always see the same thing. That's why we get the structures. For disordered regions, you don't get structures because they move. And flexible regions also move. So the question that I briefly addressed before was, is that the same? So you're in a regular three-dimensional structure, and you're in a disordered region. Is disorder just some subset of very flexible regions? And the answer is not. There's some of that, but that's not all there is. So another way in which you could go at trying to predict disorder is by looking at contact density. Well, that's very simple. So the idea here is when you have a wobble chain and the wobble chain does not form any contacts, then it can wobble around. If, however, you have a situation where sort of every part of this chain binds somewhere else, then you have a highly confined, constrained problem, and then you cannot have disorder. The more binding sites you have in this thing here, the more it cannot move around. Flexible residues are the ones that are somehow standing out, that are not constrained by binding to other regions here. All of these 6, in this particular case, I'm building a sheet again here with my wire, but they are constrained because they're closely bonded. So then the idea would be to simply predict so what I show you is the residue number, and I predict how much two residues bind to each other, the darker, the more contact is predicted. And I essentially go for this and say that regions where I have very few binding are regions that I call disorder. And that method can predict disorder. And then there's another method that has to do with the way in which we essentially look at loops or in the b-value again. Now, how can we measure disorder? And that is a problem. There are different types of unstructured or disordered regions. Some of those are such that upon binding a substrate, they adopt a regular 3D structure. So they wobble around until they find a binding partner. When they find a binding partner, they adopt a unique three-dimensional structure. Why could you imagine that this would happen? Why would you be wobbly running around and then something comes and you bind? So you would be like this, wobbly, 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 and binding, now you induce a structure. What could be the advantage of that? Any idea? Yeah?
You could be more flexible to different binding partners.
Exactly. You could imagine that you bind different binding partners with this. This is exactly what they do. So the assumption is that one mechanism why you have disorder is to bind different binding partners. Another example why you may have disorder is assume that you have a very long floppy thing. What could you do with that? You could wave. A protein doesn't really wave. They are social beings, but they don't wave at each other. Does that give you any other idea what you could do with this? Rather than with this? Well, that's essentially what she said, right? So you can bind to this, and you go and bind to something that looks different, yes. But now I'm asking about some other feature. I'm actually waving at you, nobody's waving back. No. That doesn't give you any idea? No? Yeah? Like a fly trap, yes. You could go, you reach much further, and you can possibly sense something that you could trap. But you could also just sense it. So you have a larger space that you sample, and you could sample an intruder. Again, that's another reason for disorder. There's a type of disorder that obviously is such that you do not adopt a three-dimensional structure, and the aspect of this disorder is to remain unstructured. And the idea really is that you're like the Amazon, when you get an Amazon package, the filling material. So you just produce something that is like filling material. It's sort of soft, and it's put in between other things. And again, that exists on the level of proteins. The problem with all of that is that you can also, since you bind to many things, you could aggregate to things that you're not supposed to bind. So you could do more damage. And that, in fact, is a problem, and we assume that this is one of the reasons why in the prokaryotic world there is less regulation and there's less of disorder. But there are very, very different types of disorder. But again, we cannot experimentally measure them. Here's an idea from Keith Dunker, and that idea says, well, let's just look at three-dimensional structures of proteins, X-ray structures, and let's look. There's always some residues that we actually cannot see. Cannot see means you shine, I said you take the sequence, throw it in solvent, it adopts a unique three-dimensional structure, you shine light, you always see the same thing. Very flexible regions, you don't always see the same thing. So actually you don't get a consistent signal, you don't see. And therefore the idea really was to predict disorder based on essentially that signal. Build a database. And there are many, many methods that do that, and they get a certain performance. And let's go through that. But here is something that is very important. but it's biology. I thought I would get further in the slides on the method part. Instead, I go into biology because this is one point here that I find amazing and important. So in the world of bacteria, so red and blue are two different prediction methods. And now I apply these and I look for the entire bacterium, or for this entire organism, human for instance, and I ask how many proteins. Before I said 20 to 25% of the proteins are membrane. Now I ask how many are disordered. And those are two different prediction methods, red and blue. For bacteria and archaea, this is about, you see here, around about 7 or 8 to 10% or 12%, while for eukaryotes, it's 4 times more. And you actually see there's a difference between yeast and multicellular organisms, and there's a difference between human and mouse. So the level of disorder clearly increases. Again, one reason why this is the case is what is coined more molecular recognition elements. So these are regions where you can actually bind different proteins by being disordered. This is exactly the idea that the two of you phrased. And you can essentially predict them, and you see a lot of those. Another reason, and again Keith Dunker is one of the people who pushes this idea, maybe when we consider something such as the atom of complexity, the way organisms build up complexity on the move from bacteria to eukaryotes, maybe that is done through disorder. We see that disorder correlates with the environment. Yes?
You said the level of disorders is increasing. Where is it increasing?
So eukaryotes have a much higher level of disorder. Here we have mores, eukaryotes up here, bacteria down here. So these are mores, are the regions of disorder where you bind to different proteins. And you see it's much, much, much more in eukaryotes than in prokaryotes. Maybe this is exactly so the background to the element of complexity. Maybe that's what we see. What we do see is that disorder seems to be a way in which I could induce something locally, and that is sort of 1D feature that is immensely important for building up the complexity and for reacting in certain environments. With that, given